Okay, so we just finished talking about neuromuscular junction, and now we're going to talk about electrically excitable cells. And so here we want to explain why a cell has an electrical charge difference across its plasma membrane, and in general terms how this relates to muscle contraction. Muscle fibers and neurons are electrically excitable, and their membranes, what this means is their membranes exhibit a voltage change in response to a stimulation. So the membrane has a difference in charge across the membrane. I'll illustrate that in a bit. The study of this membrane potentials within living cells is called electrophysiology. And voltage is it's kind of like a potential energy, but it's electrical potential energy. It's kind of the same thing you see like on batteries that you buy to run portable electrical devices. They have a certain voltage, which is related to the potential energy that those batteries have to do work. So our membrane in the cell membrane in the muscle cell, called the sarcolemma, is going to have a resting membrane potential of negative 90 millivolts. And so I've drawn here a phospholipid bilayer, and so to kind of explain why there is actually a difference in charge, this charge is going to be maintained by a special protein, membrane protein, called sodium-potassium pump. And this sodium-potassium pump runs on ATP, so it requires energy. And for every molecule ATP used, we're going to pump out of the cell. So this, by the way, is the intracellular fluid, and here's the extracellular on the outside. So for every ATP, we're going to pump out 3 sodium ions and 2 potassium ions. So overall, we're pumping out 3 positive charges and 2 negative charges. After a while, we're gonna end up with more positive charge outside than on the inside. Add to that, you're gonna have proteins within the cell. Now, I'm just trying to illustrate it here, and I'm doing a negative sign on there. So overall, these cells or these proteins are gonna have a negative charge. So after pumping out more positive outside than on the inside, overall we're going to have more negative on the inside and more positive on the outside. So this charge difference is what allows us to do work. Positive charges want to come across, negative charges want to go across and join up with the opposite charge, but they're kept separate by this membrane here. So there's potential to do work if we were to allow those charges to move. Overall, the charge in a muscle fiber, or the potential, is going to be negative 90 millivolts. And it's negative because this is with respect with the internal environment of the cell. Overall, the internal environment is negative, with outside is positive. If this was respect for the outside, then we would say it's positive 90 millivolts, but we're doing it with respect to the inside. Okay. So in an unstimulated resting cell, there are more anions or negatively charged particles on the inside of the membrane than on the outside, which I just illustrated as to why. These anions are going to make the inside of the membrane negatively charged by comparison to its outer surface. The membrane is electrically polarized, which means charge, and it will have a resting membrane potential when it's not actively when the membrane is not actively changing its potential, which will end up doing work within the cell. There is going to be excess of sodium on the outside in the extracellular fluid and an excess of potassium in the intracellular fluid. So we know that we have, on the outside, we're going to have excess of these sodium ions. I'll just draw a bunch of positives to indicate more of these positive charges. On the inside, we're going to have potassium ions with some of their positive charges, but remember we also have lots of negative or negatively charged proteins on the inside. Okay, So when we stimulate a muscle fiber or a nerve cell, the same general idea is going to happen here. There's going to be some other special membrane proteins. I'm going to draw one here. And this special membrane protein will only allow sodium to go through. And then I'm going to draw another special protein here that's very specific for potassium. And at this point in resting, they're both basically closed here. But if we were to stimulate the membrane in some way, this gate here can open. And when it opens, because there's a lot of sodium on the outside and less on the inside, the sodium is going to want to rush in. Okay? So the sodium ion gate opens, and when it opens, we're going to have a bunch of sodium rushing into the cell. So the sodium flows into the cell down its electrochemical gradient. Electrochemical, because there's lots of positive charge, positives don't like each other, but also just because there's a lot of sodium, we're going to run into the cell down its concentration gradient. When they do come in here into the intracellular fluid, we're going to be adding more positive charge. So remember that we started at negative 90 millivolts. So as the positive starts coming in, it overrides the negative charges. So we're going to have an increase in the value. We're going to go from negative 90 millivolts. We're going to start becoming more positive. So we're going to move upward. Okay. This process is going to be called depolarization. Okay. And so there's the term right there. So depolarization of the inside of the membrane. It's going to become, the membrane is going to be depolarized because the inside becomes more positive now. Okay. Immediately when we get to a value, say, of plus 75 millivolts, the sodium gates are going to close. So that doesn't allow any more sodium to go in or out. And the potassium gates are going to open. When the potassium gates open, we have a lot more potassium in here. The potassium gate cannot allow sodium to go through. It's specific for potassium. So when we do that, the potassium rushes out. So now we're having positive charges leave out of the membrane. And what that's going to do is it's going to bring back now, because we're going to lose positive charges from inside. It's going to bring back down the electric potential back down to negative 90 millivolts. Okay, so this process is going to be called repolarization. Okay, now don't know why this drew an extra line there for me, but there you go. So we have now the potassium rushing out of the cell. The reason why it rushes out is because you have a lot more potassium in here, but you also have lots of positive charge in there now. So the positive charges don't like each other, and the potassium wants to spread out as much as possible. So the potassium is going to move down its concentration gradient from higher concentration and higher positive charge down to a lower value, and that again brings back down the or brings the inside of the cell or the intracellular fluid to a more negative value. So this loss is going to be called the potassium ions, which returns the membrane potential back to negative. Is going to be called a repolarization. This quick up and down shift going from depolarization to repolarization, when this occurs because, again, the potassium gates open up, is going to be called an action potential. Okay. So when this occurs, by the way, these gates here are going to be voltage gated. And that means the gates are sensitive to changes in voltage. So when the voltage changes, because of some stimulus, that causes the sodium gate to open. Okay? And the voltage is changing, and it changes to plus 75 millivolts. That causes the sodium gate, sensitive to that change in voltage, causes it to close. The potassium is also voltage gated, and it's sensitive. So at plus 75 millivolts, it opens up and allows the sodium to rush out. That's called repolarization. As this occurs right here in this little area, you're going to have more of these voltage-gated membrane proteins, both specific for sodium and another one specific for potassium, are going to occur along the membrane. So since the membrane potential is changing here, This is going to get other gates to open and close, just as we explained before, and that's going to basically propagate itself or move along the membrane. And that process then of a moving action potential along the membrane is going to be called an impulse. So let's just go ahead and read off the notes here. It says that resting membrane potential is seen in a waiting excitable cell, whereas an action potential is a quick event seen in a stimulated excitable cell. An action potential is going to perpetuate itself, which I was talking about just a little bit ago, down the length of the cell's membrane. So an action potential at one point causes another one happening immediately in front of it, which triggers another one further along the way. So this wave of excitation is going to be called the impulse, which I drew a while ago, back over here. So there's your impulse right there moving along the membrane. So now that we kind of have an idea of how nerve cell interacts with a muscle cell at the neuromuscular junction, we can now understand when toxins affect the system and can cause paralysis, where you can't move your muscle. So the toxins can interfere with specific function of the communication between the neuron and the muscle cell. And when that occurs, you can get paralyzed. So just as kind of a review, I placed that picture of the neuromuscular junction up there, just to remind you that right here we have the synaptic knob here, and there you have your synaptic vesicles that contain the neurotransmitter acetylcholine. And then here's the synaptic cleft right here. When the nerve impulse comes down here, it's going to get the acetylcholine to be released into the synaptic cleft, and then the acetylcholine finds these receptors here. These receptors then are going to grab onto the acetylcholine and then cause membrane potential changes to occur here, which sets off an impulse along the membrane of the muscle cell, which this down here represents the muscle cell. So we can see how toxins can influence this in a negative way. For example, some pesticides are or contain cholinesterase inhibitors. Remember from earlier in the lecture that there is an enzyme within the structure here that breaks down the acetylcholine, which is necessary. If you don't get rid of the acetylcholine, then your muscle is going to keep contracting. So the acetylcholine esterase would break down the acetylcholine and get the muscle to stop contracting. What happened here is the pesticides bind to acetylcholine esterase and then prevent it from degrading acetylcholine. This would cause a condition called spastic paralysis, which is a state of continual contraction of the muscles. And since some muscles that control breathing, control the opening to your airway, can go into spasm, you could possibly suffocate, so that's not good. There's another disease called tetanus or lockjaw caused by a bacterium called Clostridium tetani, and the way this one also causes spastic paralysis is that there is compound glycine found in the spinal cord, which normally stops the motor neurons. Let me go back here. So normally stops the motor neurons, which the motor neurons are the ones that tell the muscle to, or send message to muscle to contract. This glycine helps keep the motor neurons from sending unwanted signals to the muscles to contract. So what tetanus toxin does is it blocks the glycine release in the spinal cord, and this causes overstimulation and spastic paralysis of the muscles. So basically the motor neuron keeps sending messages to the muscle cell to continue to contract. And so opposite, or the complete opposite of spastic paralysis, is flaccid paralysis, where the muscle cannot contract. So in flaccid paralysis, the muscle is limp and can't contract. So there's a poison called curare that is going to compete with acetylcholine. So this curare competes with acetylcholine. So imagine we release acetylcholine, But in this area here, you have the carrier there going and bonding to the acetylcholine receptors, little blue structures in our model here. And so if they get in the way there, then the acetylcholine can't go and bind and get the muscle to begin the contraction. So this causes flaccid paralysis. There's plant poisons in South America that the natives used to poison, to use as poison on their blow dart guns. So this would cause that types of paralysis. And then there's botulism. This is going to be a type of food poisoning caused by neuromuscular toxins secreted by another bacteria in the genus Clostridium, but this one is Clostridium botulinum. And here, the toxin of Clostridium botulinum blocks the release of acetylcholine. So you can't even get acetylcholine to be released. So if you can't release acetylcholine into the neuromuscular cleft in here, then the receptors won't receive it, and you will not begin or get a contraction. By the way, this toxin is used in Botox cosmetics, so they literally paralyze some of the muscles around the face with some injections there. So now we're going to move to section 11.4, which is the behavior of skeletal muscles. And we're going to go through each of these objectives. We're going to explain how nerve fiber stimulates skeletal muscle. We're going to explain how stimulation of the muscle fiber activates its contractile mechanism, and then explain the mechanism of muscle contraction. How does the muscle actually contract? And then we're going to explain how muscle fiber relaxes, and then we're going to talk about the relationship between the force produced by muscle contraction and the length of the muscle itself and the length of the microscopic structures within the muscle called the sarcomeres, which we covered earlier. So muscles are going to contract in four phases. One is excitation. Then we're going to go through a situation where we combine or connect, make a connection between excitation, which is the action potential on the membrane, and connect that to the contraction. So that coupling refers to connecting the two processes, the excitation to the actual contraction. So that's another phase. And then we're going to go through the actual step of contraction. How does the muscle actually shorten and produce a force? And then relaxation. When stimulation ends in the muscle fiber, how does the muscle fiber go about relaxing? So let's start with excitation, and here your objective is to explain how a nerve fiber stimulates a skeletal muscle fiber. So here's our nerve fiber, which is basically the end of a neuron. So here you have your nerve signal, your impulse coming down here. When that impulse comes down here, it's a moving action potential, just like in the muscle cell. When it gets down here, there's going to be, in the membrane, in the synaptic knob area, there's going to be a voltage-gated calcium channel. So these calcium gates are sensitive to voltage changes and only allow calcium to move. So here calcium will rush in because there's more concentration of calcium ions on the outside. When that calcium comes in, it's going to get these synaptic vesicles, which contain acetylcholine, to be released into the synaptic cleft. So that's shown over here. The vesicles are releasing by a process of exocytosis. There are the little acetylcholine molecules going and binding to these acetylcholine receptors. The acetylcholine receptors, by the way, are another kind of gate called a ligand, not a voltage regulated gate, but a ligand regulated gate. So these are ligand gated proteins. So when the two ligands, in this case a ligand is a molecule, these ligands are acetylcholine. When the two of them bond, they're going to get or activate the receptor to become permeable or allow sodium to move in and potassium to move out, kind of like a combination of both separate sodium and potassium gates. So we'll go to that stage there. So here we have, again, we're still in excitation. So here we have the acetylcholine molecules binding, or ligands binding to the receptor. When they do that, that's going to open the gate here and allow the sodium to rush in. So the sodium rushes in, and it's going to, and then so that changes the membrane potential there, and then it's going to allow the potassium to rush out, repolarizing the membrane. So we have the depolarization and repolarization of the membrane. So this is going to change our, when the sodium enters, is going to change from negative 90 millivolts to plus 75. And then when the K exits, we're going to go back to negative 90. So when we come back over here, this is going to be our depolarization, and then this would be our repolarization. This quick shift here is going to be called the end plate potential there within the synaptic cleft. Now, as we get near the ends of the neuromuscular junction here, nearby along the sarcolemma, you're going to have a specific voltage gated. So here's your ligand gated, but here's specific voltage gated. So as the membrane potential changes here in the end plate area, end plate potential, that change in voltage now is going to activate sodium gates to open up. So the sodium gates open up, we rush in, we're going to get a depolarization right here. So this would cause depolarization. And then once we get to about plus 75 millivolts, then the potassium gate opens up and allows positive charges out. That gives us our repolarization. And then this action potential that's occurring right here is going to stimulate another action potential over here, and then another one nearby. And so as that continues down the sarcolemma, which is the membrane of the muscle fiber, we would call that our impulse. Okay. Now we're going to come to the coupling of excitation to contraction, or what joins the two. So here, this objective, you need to explain how stimulation of the muscle fiber activates its contractile mechanism. So to do that, I wanted to review a little bit of the microscopic anatomy of the muscle fibers. Here's our muscle fiber. The yellow represents your sarcolemma there. Recall earlier, you can't see them because I have it pretty small here, but there were like these pores along the sarcolemma, and those pores enter into those purple tube-like structures that are called T-tubules or transverse tubules, and they're associated with the sarcoplasmic reticulum, which is this blue network of membranes in there. And within there, within the sarcoplasmic reticula, calcium ions are stored and kept away from the cytosol, or the fluid of the membrane. So, and they're kept away from these fibrils, myofibrils here that contain the proteins for contraction. So let's continue here. We just finished talking about the impulse coming in the excitation. The excitation caused the release of the neurotransmitter, and then we had our end plate potential, which then stimulated the impulse to occur along the sarcolemma. There's one of your pores in the membrane. So here the impulse is going to go down the T-tubule, and as it goes down the T-tubule, here is your endoplasmic reticulum, that in blue, which is where the calcium is stored. So those little white spheres are the calcium. As the impulse comes in here, then the calcium is going to get the calcium to be released into the cytosol. So that's shown here on the picture on the right. So the calcium gets released from the ends of the endoplasmic reticulum, which, by the way, recall they're called terminal cisternae. And they get released there. So now we're going to have a bunch of calcium in the cytosol. So those myofibrils, which contain those filaments for contraction, are going to be exposed to the calcium. So let's move ahead now. And still within the excitation-contraction coupling, what's going to happen here is that calcium is going to go to those thin filaments where, recall, we had the tropin and tropomyosin complex, and those the tropomyosin actually covers the active sites along the actin units there. And so when the calcium binds there, it's going to cause this to change shape. When it does, the tropomyosin moves out of the way of the active site. So that's going to allow the myosin, which is the basic units of the thick filaments, going to allow the myosin ability to bind there. And so at this point here, we're going to progress into the contraction phase. So we're going to kind of follow this little diagram that takes us through a cycle. So we're going to start with number 10 here, step 10 in this process. And so what's going to happen here is ATP is going to come and bond with the myosin head, And so if we come back over here, if you looked at the shape of the myosin head right here, the myosin head seems to be flexed at this point. So when the ATP bonds to it, and then the ATP is hydrolyzed, it goes from adenosine triphosphate to adenosine diphosphate, because we're going to break off one of the phosphates. So there's your other phosphate there. That's going to cause the head of the myosin molecule to extend outward, to become loaded, kind of like a spring. And what's going to happen after that is the myosin head then is going to form a connection to the active site right up here. And when it does that, that's going to be called a myosin-actin cross bridge, because there's like a cross connection to there. Okay? Now, moving on to the next step, which is right here on step 12 here. When the connection is made there, then the myosin head is going to release the ADP and the phosphate, which is going to probably go back to the mitochondria to be turned back into ATP. But when it releases the ADP and the phosphate, the myosin head is going to move back to its original flexed position. And when it does so, it's basically going to pull the thin filament along with it. So that's going to be referred to as the power stroke. Okay? And so then we'll repeat the cycle again, coming to step 13 here, where we're going to bind to a new ATP. When the new ATP hydrolyzes, again, it's going to, the myosin is going to flex outward, and we repeat the process over again. Okay, so this can actually happen. Let me move back. I went ahead there. This can actually happen up to 5. We can get 5 of these power strokes per second, and each of the power strokes would require the use of 1 ATP. So now let's look at relaxation. So here your objective is to explain how a muscle fiber relaxes. Okay, so first of all, we want the nerve stimulation and the acetylcholine release to stop. So we stop the nerve impulse from coming in. That will stop the release of the acetylcholine. And the acetylcholine esterase, the ACHE, is going to break down the acetylcholine. So in the picture on the right, we stopped releasing acetylcholine. We have an enzyme called acetylcholine esterase, which goes and breaks down the acetylcholine. Okay? And that will basically close off the gate here and stop sodium and potassium from moving down their concentration gradients. So basically the stimulation stops. Now then, going back over here, we are going to pump the calcium ions back into the sarcoplasmic reticulum by active transport. So that's going to require energy. The calcium is going to bind to a protein within there, which will help store it. And let's see, if you remove the calcium away from there, what's that going to do to the tropomyosin? It's going to cause the tropomyosin to move back and cover the active sites that we saw earlier. So when we remove that calcium, the tropomyosin will reblock, as shown right here, and the myosin heads can no longer form those cross bridges. So then your muscle's going to relax at that point. There's a final another topic to discuss here, which is the relationship between the length of your muscle, or at the microscopic level, the length between the sarcomeres. So recall over here, each of these from here to here, from each Z disc, is the functional unit within there, and this is very tiny unit within a long muscle fiber. So on the order of micrometers, which is millionth of a meter. Okay, but it turns out that you can actually change that distance between there, and that's essentially what happens when you contract. The distance between these Z discs decreases, and that overall will, when that happens to every sarcomere down the line altogether at once, that causes the muscle fiber to shorten. With all the muscle fiber shortening, the muscle shortens and produces a force on the structures that the muscle is trying to move. It turns out that there is a relationship between the length between one disc, Z disc, to another, and the amount of force that can be produced by the muscle. Now, because the muscle's force is produced by pulling on protein fibers, in physics we call those forces on cables and ropes, that stretching force, we're going to call that tension. So tension is basically a force. Kind of like if I hung a 50-pound weight from a rope, the 50-pound weight's hanging from the rope, and there's going to be 50 pounds of tension within the rope. The point here now is that if we start with the distance between the sarcomeres way too shortened, so we have overshortened distance, before we stimulate the muscle to contract, we're going to get a weak contraction. And the reason for this is that the thick filaments are going to butt up against the Z disc. So imagine here, if these Z discs were actually closer right here, okay? And then we start our contraction. And those little thin filaments, the thin filaments get pulled inward by the myosin heads during the power stroke. The Z discs are going to just basically bump into those thick filaments, so they can't move very far, so you're not going to get a very good tension in the muscle. Now, if we're too stretched before stimulation, you're going to get a weak contraction, but for a different reason. Imagine if we pulled these Z discs outward. That's going to take those thin filaments and stretch them out or move them out further. So we're going to have a reduced overlap between the thick and the thin filaments. So you're going to have a reduced ability for those cross bridgings to form. And it's kind of like trying to pull yourself up from a rope, but you can only reach the rope with your fingertips. You're not going to be able to get much of a grip on there, so you won't be able to pull yourself up on the rope. So it's kind of a similar situation. So what turns out here is we have a optimum resting length. So this optimum resting length is going to produce the greatest amount of force. And so there's an optimum distance. And so this optimum distance is going to be maintained by your nervous system, which is constantly producing a tone in your muscle or some partial contractions that will help keep your muscle at the right length. I don't know why this thing continues to keep forwarding on me. There's a little glitch in my equipment here, but let's just try to move forward here. So this graph here is going to show that relationship here. Up here we have part of a myofibril here, and here's what happens if your sarcomere are too short. This is part of a Z disc, part of the myofibril here. Here we're only demonstrating with one thick filament in the middle, and there's your thin filaments on either side. So remember, those are the myosin heads that are going to grab onto the thin filament and during the power stroke pull them inward. Well, if we start with the distance way too short, and down here on the bottom axis is the sarcomere length in micrometers. If it's too short here, you're not going to be able to pull it much further in, so you're going to get a very reduced tension. But as you start to increase the length, move the distance of the Z disc outward, which is going to pull the thin filaments out with it, then you're going to be able to get, as the graph shows, you're going to get greater and greater tension until you get to a optimum point here, an optimum distance. That optimum distance is indicated by this blue shaded area here, and that distance is between 2 and 2.25 micrometers in distance. Now, so there you can see there's a good amount of overlap. The cross bridging can occur quite nicely right here, and right here, and right here, and right here. So when the cross bridging occurs, power strokes occur, we can pull those discs in and get a good tension in the muscle. Now, if we stretch the muscle out further, and we stretch the Z disc further, eventually we're going to lose that optimum range, and so the tension begins to drop here as we increase that distance. And this shows why. If you start out too far, there's hardly any overlap right here. And as I mentioned, the analogy would be trying to grab, you can barely reach a rope and you're trying to grab it with your fingertips. You're not going to be able to get much of a connection there. So very, very minimal amount of cross bridging can occur. So it's going to be hard for the Z disc to be pulled inward. So there's your reduced force. So now comes up the topic of rigor mortis. And rigor mortis is what happens when an animal dies, like a human. Human's an animal as well. The muscles become hardened and stiffen the body, and this is going to happen within 3 to 4 hours. Now, because we understand how muscle contraction works, what causes rigor mortis will make more sense. Okay, so What's going to happen is, remember the blue network of sarcoplasmic reticulum that holds the calcium and keeps it away from the myofibrils where the troponin is, so it keeps it out of there. Well, the sarcoplasmic reticulum begins to break down. When it breaks down, the sarcolemma is going to release calcium. It's going to enter the cytosol. The calcium will activate the cross-bridging we talked about. What's going to happen here is the muscle is going to contract, but it cannot relax. Wow, that makes good sense. So what then? Why can't it relax? Well, in order to relax, remember when we were going through the power strokes, those power strokes are going to require the use of ATP. But when cells die, they don't produce any more ATP. So without any ATP, the myosin head can't release from the thin filaments. Okay? So the muscle remains contracted. And this is going to continue to remain contracted until the myofilaments begin to decay. So once bacteria starts to act and break down, then eventually the body loosens up again. At that point, the body of the animal may not be smelling too good at that point. But this right here, last point here, is rigor mortis is going to peak. It'll start, as we said earlier, it'll start about 3 to 4 hours after death, and then maximum rigor mortis about 12 hours after death. And then it'll begin to diminish 48 to 68 hours afterwards due to the decomposition of the protein fibers themselves.